In this talk, I present our laboratory's approach to assessing cerebrovascular health. I am therefore drawing upon the experience garnered over more than 20 years by a considerable number of us at Thornhill Research, the University of Toronto Health Network, and Toronto Western Hospital, led by Drs. Joseph Fisher and David Michaelis. In most slides, the figures are taken from our published papers, and the references list the names of the investigators involved. It is thanks to all that I am able to assemble the material for this talk. To begin, recall some of the basic physiology of the cerebral circulation. As this picture humorously illustrates, the adult human brain of about 1.5 kilograms uses roughly 20% of the oxygen supplied by breathing, in a blood flow that takes about 15% of the cardiac output to do so. And as this picture illustrates, the cerebral blood supply must not fail. Many mechanisms are involved in the protection of cerebral blood flow, and I reviewed them in another talk about the control of cerebral blood flow. Nevertheless, a brief review is necessary for understanding how cerebrovascular health may be assessed. Blood flow is routed to brain regions by the smooth muscles of the parenchymal arterioles. The large peel arteries on the surface of the cortex contain multiple layers of vascular smooth muscle cells. These peel vessels branch into penetrating arterioles containing a single layer of vascular smooth muscle cells. Regional blood flow in the normal and ischemic brain is controlled by arteriolar smooth muscle cell contractility and by capillary pericytes. A global CO2 stimulus affects all cerebral smooth muscle cells at once. As shown on the left, cortical blood flow is distributed bilaterally from the circle of Willis via major intracranial arteries, the middle cerebral artery MCA, the anterior cerebral arteries ACA, and the posterior cerebral arteries PCA. These feed their regions of the cortex via penetrating arterioles to the capillary network supplying the tissues. On the right, this distribution system can be viewed using a hydraulic analogy as a flow resistance network, with resistance controlled by changing the vessel diameter via the smooth muscle lining vessels. Note the high resistance of the major supply arteries, producing the 20 to 30% of the total pressure loss as blood transits the brain. When an increase in arterial PCO2 occurs, hypercapnia, and global blood flow increases, this high resistance produces a drop in overall cortical perfusion pressure. Consequently, blood flow in those regions unable to vasodilate sufficiently may decrease, in contrast to blood flow in regions with robust vasodilation, which will increase, appearing to steal the lost flow. And this observation is known as the steal phenomenon. What can go wrong with the supply of blood to the brain? And how does the cerebral circulation cope? Here is one example, a stenosis in part of a vasculature. And these pictures show the ways in which the cerebral circulation may adapt. Adaptation to maintain blood supply can take two forms: downstream vasodilation to reduce the overall resistance to flow, and/or the development of alternate pathways, collateral vessels. In A, a normal vasculature is shown on the left, and on the right, a vasculature with a stenosis, where normal blood flow is maintained by a collateral replacing the stenotic vessel. In B, the left vasculature has normal blood flow maintained by vasodilation, using up the vasodilatory reserve. The right vasculature has a narrow collateral, insufficient by itself to maintain normal blood flow, and it too vasodilates. In C, the left vasculature stenosis is severe, and blood flow is reduced despite vasodilation. For the right vasculature, the collateral is too narrow, and all vasodilatory reserve has been used, so it too has a reduced blood flow. In both A and B situations, blood flow is maintained. Only in the more severe case C does blood flow decrease. Consequently, only condition C can be detected by measuring resting blood flow. To detect condition B requires measurement of vasodilatory reserve, or the ability of the vascular to vasodilate. The latter assessment requires the application of a test vasodilatory stress and measurement of the resulting increase in blood flow. This assessment is cerebrovascular reactivity, CVR, the ability of a vessel to increase blood flow when a vasodilatory stress is applied. The measure used to assess cerebrovascular health is cerebrovascular reactivity, CVR. It is a vasodilatory stress test that measures the ratio of the change in blood flow per change in vasodilatory stress. For this test, how is blood flow measured and what is the vasodilatory stress used? Cerebral blood flow can be measured in many ways, some illustrated here. Two of the most popular and easiest to implement are transcranial Doppler, TCD, for measuring the blood flow velocity in a major artery like the middle cerebral artery, and the recording shown on the upper right is the beat-by-beat changes in blood flow velocity detected from ultrasound measurements made via the right and left temporal windows. To obtain measures of blood flow throughout the brain, several techniques can be used, and magnetic resonance imaging is a convenient and easily implemented method, and the blood oxygen level dependent signal, BOLD, a popular surrogate measure of blood flow changes. Indeed, it is the measure that we use. The lower left figure shows a comparison of arterial spin labeling, ASL, and BOLD measurements of changes in cerebral blood flow. The vasodilatory stress used to measure CVR involves overwhelming the normal regulatory systems that control cerebral blood flow. And there are three aspects to this control listed on the left. The first is termed autoregulation, which attempts to maintain blood flow constant in the face of changes in arterial blood pressure. The second is termed neurovascular coupling, whereby blood flow to a region is increased during increased metabolic and neuronal activity. And finally, the respiratory chemoreflex control of breathing attempts to keep oxygen supply and carbon dioxide elimination constant in the face of environmental changes, in order to control the arterial blood content supplying the brain. Consequently, there are several ways of providing a vasodilatory stress by stressing these three control systems. However, the most practical stressor is carbon dioxide, CO2, which acts to produce vasodilation by its action on vascular smooth muscle. Here is a pictorial overview of the vasodilatory effect of carbon dioxide, showing data assembled from various sources and experimental techniques in review from the Ainsley Laboratory. These observations at various points in the vascular tree show that vessel diameter increases at all levels of the cerebral vasculature. In A, the precapillary and capillaries. In B, a Peel vessel. In C, the middle cerebral artery. And D, the internal carotid supply artery. Cerebral blood flow changes with blood gases, oxygen, and carbon dioxide. And this graph shows transcranial Doppler, or TCD, measurements of middle cerebral artery blood flow velocity. As the arterial partial pressure of carbon dioxide increases from 25 millimeters of mercury, hypocapnia, to 55 millimeters of mercury, hypercapnia, at isoxic tensions of both 50 millimeters of mercury, hypoxic, and 150 millimeters of mercury, hyperoxic. The middle cerebral artery velocity increases in a sigmoidal fashion. Comparing the solid and dotted line fits of these sigmoids, it can be seen that hypoxia enhances the response to CO2. The sigmoid fit to these data result from the vascular limitations of vasoconstriction in hypocapnia and vasodilation in hypercapnia. Note too that blood pressure, the blue lines, was also measured in these experiments and acts as a confounder because changes in blood pressure directly affect blood flow. In this individual, blood pressure was constant until a threshold partial pressure of CO2 was reached around 45 millimeters of mercury. Above, blood pressure and cerebral blood flow increased linearly with the partial pressure of CO2. The constant blood pressure below the threshold enabled the sigmoid vasodilation response to CO2 to be seen, unaffected by blood pressure changes. Above the threshold, however, the increasing blood pressure increased the cerebral blood flow proportionately. The picture at the right is a brief overview of how CO2 changes vessel diameter and the resistance to blood flow. Carbon dioxide disrupts the regulation of intracellular hydrogen ion concentration in smooth muscle cells and changes smooth muscle tone via mechanisms involving changes in calcium and potassium ion intracellular concentrations. An older method that was used to increase CO2 was an injection of acetazolamide, a carbonic anhydrase inhibitor, which hinders the elimination of metabolically produced CO2 and thereby causes a rise in the arterial partial pressure of CO2. However, acetazolamide is invasive and its effectiveness varies considerably among individuals. Moreover, there can be adverse side effects, and its action cannot be easily reversed. Another method of increasing CO2 is the inhaling of a fixed inspired CO2 concentration, as illustrated on the left. However, as shown in the middle graph, there is no ability to execute a step change in CO2 and no regulation of oxygen, which itself is vasoactive. Moreover, each test will produce different arterial partial pressures of carbon dioxide and oxygen, depending on an individual's variation in their breathing response to these changes. In addition, as the rightmost graph demonstrates, everyone has a different end-tidal to arterial gradient for CO2, so that end-tidal measurements do not measure the actual arterial partial pressure of CO2, which is the actual vasodilatory stimulus. Hence, I include the quotation from Fisher that the main concern is that declaring the precision of a fixed inspired concentration of CO2 is misleading. It does not, as implied, translate to precise control of the actual vasoactive stimulus, the arterial partial pressure of CO2. the inherent test-to-test and therefore subject-to-subject variability precludes clinical application of findings. Yet another method of increasing carbon dioxide is to hold one's breath. Here is a picture of the sequence of events in a breath hold test on the left. And on the right, note that during a breath hold, the partial pressure of carbon dioxide increases and that of oxygen decreases. And so this test applies a mixed vasodilatory stimulus. Moreover, even with a fixed breath hold time, the change in the arterial partial pressures of both carbon dioxide and oxygen depend on an individual's metabolism, and so the stimulus may vary considerably between individuals. A further consideration is that the vasodilatory stress may be insufficient to reveal pathological conditions. Our experience suggests that at least a 10 millimeter rise in the arterial partial pressure of carbon dioxide is needed to show pathology. If CVR measurements are to be of use in a clinical setting, they must be made with a standard stimulus so that comparisons may be made between individuals to distinguish deviations from normal, as well as comparisons in the same individual over a period of time to assess pathological changes over time or with treatment. Sequential gas delivery combined with a precision gas blender and appropriate software provides control of arterial partial pressures of carbon dioxide and oxygen independent of an individual's breathing pattern and independent of each other. This methodology, therefore, is capable of meeting the requirement for a standard and repeatable stimulus. The graphs below illustrate the capability of this methodology, and further discussion of the various methods used to provide a carbon dioxide stress is available in the references noted. Here we take a quick diversion to explain how sequential gas delivery works in order to control arterial blood gases. And please note that although pictured as physical reservoirs here to illustrate the principles involved, their actual implementation is made in the Respiract software. Now, as shown, a patient breathes normally through a mask connected to the breathing circuit. One-way valves, shown as red, ensure that inspired air comes from the inspiratory reservoir and expiratory air goes to the expiratory reservoir. And the crossover valve, shown in yellow, which has a slightly higher opening pressure than the one-way valves in red, only opens when the inspiratory reservoir is empty. So pictured here is the situation at the end of expiration, when expired air in dark blue, labeled as gas 2, fills the expiratory reservoir, and the gas used for targeting arterial PCO2 and PO2, gas 1, fills the inspiratory reservoir to a specified volume. During inspiration, the specified gas mixture destined to control arterial blood gases, gas 1, comes from the inspiratory reservoir at first via the red one-way inspiratory valve. As inspiration continues, the inspiratory reservoir is emptied, and gas 2 is inspired from the expiratory reservoir via the crossover valve. Now this gas has already been used for gas exchange with the blood in the previous breath and is therefore neutral with respect to controlling arterial blood gases. This picture summarizes what has happened during the inspiration. starting at the end of expiration on the left and showing how gas 1 enters the functional residual capacity and the alveolar space where gas exchange with pulmonary blood occurs. And at the start, A, the FRC and airways are filled with expired gas. During phase B, gas 1 is inspired, eventually collapsing the gas 1 reservoir, so that during phase C, expired gas 2 is sequentially administered via the crossover valve. The result is that only the specified volume of gas 1 with the specified mixture reaches the gas exchange space. And recall that gas 2 has no effect on gas exchange because it's already been used. So in this way, the respirator can control arterial blood oxygen and carbon dioxide independently and independent of the patient's breathing pattern. We have conducted CVR testing for over 20 years, both experimentally and clinically, and found them safe and well tolerated, with no adverse events encountered. The picture on the right shows our gas delivery system connected to a subject in the MR environment. Testing consists of the application of a precise, reproducible carbon dioxide vasodilatory stimulus, while maintaining oxygen at its normal resting value, coupled with bold MRI to measure changes in blood flow. The test is non-invasive, requiring no radiation or injections. Bold MRI measurement of blood flow changes have high spatial and temporal resolution, showing detailed locations of lesions and permitting speed of response measurements. Here is the most basic calculation of CVR. The change in blood flow divided by a change in stimulus. That is, a change in the bold signal divided by the change in the partial pressure of CO2. And in this case, it's based upon two partial pressures of 40 and 50 millimeters of mercury, PCO2. Blood flow is measured as the change in the bold signal, and with the respirator, the change in end-tidal PCO2 is the change in arterial PCO2, the vasodilatory stimulus. Our experience measuring CVR has taught us some important lessons. They are set out in detail in the stated reference, and I will briefly review some of them here. First, the graph shows changes in the bold response over a range of CO2 for 3 voxels. It is apparent that the responses from the 3 example voxels differ, so that the slope of a 2-point line, such as provided by a boxcar CO2 sequence, will depend where the range of CO2 is chosen. Indeed, it is clear that CVR depends on whether the CO2 range is in the hypocapnic or the hypercapnic range. Another important consideration is that CVR depends on the magnitude of the stimulus, as the top row of maps demonstrates. It is apparent that it must be sufficient to show pathology. We find that to reveal pathology reliably, an increase of at least 10 millimeters of mercury PCO2 is needed. In addition, the choice of baseline is important. Comparing the top row CVR maps with a 40 millimeter mercury PCO2 baseline with those of the bottom row with a 30 millimeter mercury PCO2 baseline and the same 10 millimeters of mercury change demonstrates the effect on CVR. Our view initially standardized all patients at a baseline of 40 millimeters of mercury. However, we soon realized that this was incorrect. The baseline should be chosen at the normal resting PCO2, because this is where vascular tone is at its regulated value. This consideration is especially important for comparing CVR between subjects. This is the experimental paradigm we use. The graph shows the whole brain average bold signal, shown in purple. The graph also shows the end tidal partial pressure of carbon dioxide in blue, resampled to the TR of the bold signal. We use bold to detect blood flow changes because it is easily implemented and has the necessary spatial and temporal resolution. We assume that changes in metabolism and blood volume do not impact the bold signal significantly enough to confound the CVR measurement. Note that the test does require a voluntary hyperventilation to lower the partial pressure of carbon dioxide before the ramp increase, but otherwise the subject breathes freely. The step portion stresses the vasculature from resting levels to 10 millimeters of mercury higher, PCO2, quickly enough to detect speed of response effects. The step increase in the partial pressure of carbon dioxide is set to be from the resting partial pressure to plus 10 millimeters. This increase, based upon experience, is sufficient to uncover pathology. The ramp portion stresses the vasculature from maximum vasoconstriction to maximum vasodilation slowly enough to avoid speeds of response effects. Before continuing to show voxel-wise maps of CVR, it is important to understand that the carbon dioxide stimulus is applied to the whole brain vasculature, which is a distributed network, and its anatomic structure dictates regional interactions of perfusion pressures. Recall, too, that the supply arteries to the brain have substantial resistance to flow. So as global carbon dioxide increases, vascular regions vasodilate and flow overall increases. Consequently, flow redistributes, partly according to the ability of the vessels to vasodilate in a region. However, this flow redistribution causes changes in the distribution of perfusion pressures in the vascular network. Why? Because the brain's supply arteries offer a significant flow resistance. Any increase in global flow results in a decreased downstream pressure. We therefore conclude that both the interaction of the changes in pressure distribution and the vasodilatory ability of a region determine the blood flow response in that region to a global carbon dioxide stimulus. Here are examples of axial brain slices showing maps of the distribution of color-coded CVR values. Positive CVR is yellow to red, and negative CVR is light blue to dark blue. On the left, a healthy individual, and on the right, an individual with cerebrovascular pathology, so that the CVR map shows regions of blue, where the CVR is negative and steel is occurring. The ability to produce a step change in PCO2 enables a measurement of the speed with which the vasodilation occurs. On the left, the time course of the bold response to CO2, the black lines, in 2 voxels are shown, the top green response slower than the bottom red response. A regression of the step change data to obtain CVR contains all the points measured as the response increases from baseline to full. And this inclusion decreases the CVR for slower responses, as shown in the graphs on the left, where the slow green response regression line in black is a lower slope than the steady state response line in green. The fast red response shows no such difference in slope. From the appearance of the response time courses shown on the left, it would appear that the response could be fitted by an exponential rise function. Indeed, this fitting is done, and the time constant tau in seconds is taken as a measure of the speed of the vascular response. Here is an example axial map of tau on the right from the STEP CVR data shown on the left. As the tau map shows, the steal is light blue and fast. Steal by the healthier voxels is immediate in these unresponsive regions of the cerebrovasculature. Even some regions with positive CVR are nevertheless slow, as shown in the tau map's green regions. So far, the maps we have looked at have been calculated using the STEP data that provides 2 levels of PCO2 stimulus. The RAMP data can also be used to measure CVR, as well as other aspects of cerebrovascular reactivity, as we shall see later. To compare with the STEP data, RAMP CVR is measured over the same PCO2 range, from resting to plus 10 millimeters of mercury. Since the RAMP change in stimulus is slow, there is time for differences in the network pressures and vasodilatory responses to reach equilibrium. Comparing axial maps of CVR calculated from the STEP portion of the sequence, where end-tidal partial pressure of CO2 increased by 10 millimeters of mercury, with a portion of the RAMP data from the resting end-tidal partial pressure to plus 10 millimeters of mercury, illustrates the point made earlier, that slow responses produce a lower CVR. When the vasculature is given time to respond so that the network redistribution of flow is complete, as in the RAMP, then the regions of steal are considerably smaller. Finally, consider the question of whether the CVR maps observed for a patient are normal or not. The only way to tell is to compare the patient's map to one assembled from an average of maps from healthy individuals. And that can only be done if the CO2 stress and the MRI bold measurement is the same for all, which rules out using breath hold or a fixed inspired CO2 concentration. Here is the procedure as illustrated. Assemble a number of CVR tests in a healthy cohort of volunteers. Co-register the CVR maps into standard space so that a mean and standard deviation of CVR can be calculated for each voxel. Then, any patient map obtained from a CVR test with the same stimulus and MR measurements can be registered into standard space and the Z-score calculated for each voxel to obtain a Z-map displaying difference from normal in standard deviations. Here is the Z-map for our example patient, with a CVR map showing steal. The Z-map shows that overall CVR is lower than normal, with the steal regions well below normal. Again, I emphasize that Z-map comparisons can only be made if the CO2 stress and the MRI bold measurement is the same for all in the healthy group and the patient, which rules out using breath hold or fixed inspired CO2 concentrations. As an alternative to simple regression measurement of CVR, transfer function analysis, TFA, provides similar information using a different approach. This is a standard signal analysis engineering technique. TFA uses the entire stimulus sequence of both step and ramp changes. It calculates the relationship between a stimulus signal, carbon dioxide in this case, and a response signal, the bold signal. It does this calculation in the frequency domain, resolving the two signals into their Fourier series of component sine waves. The resulting measurements are gain, which corresponds to CVR, phase lag, which corresponds to tau, and coherence for each component frequency. The resulting three measures are displayed as spectra, such as that from an example voxel spectrum shown on the left. For the bold response to CO2 test, I chose to map the gain, phase, and coherence at a particular low frequency where the coherence was high. And the middle graph shows that chosen single frequency and illustrates the gain as the ratio of sine wave amplitudes and the phase lag as the left shift in the response sine wave. Here is an illustration of transfer function analysis of two voxels where bold measures blood flow in percent change and the vasodilatory stimulus is two square wave changes in PCO2, the black traces. The left voxel, A, shows a negative bold response, while the right voxel, B, shows a positive bold response. The spectra for gain, phase, and coherence are shown below these traces. To provide a specific value for gain, phase, and coherence maps, the values at a frequency of 0.01 Hz were chosen where the coherence was high. These very low frequency values can be viewed as single sine waves, as shown on the right, the top for voxel A and the bottom for voxel B. Examining these sine waves, you can see in the upper graph that the bold response sine wave begins to increase before the PCO2 sine wave begins to increase. In other words, the bold response leads the PCO2 stimulus by almost 5 radians or 180 degrees. The phase is positive, signifying that the bold response is negative. In the lower graph, the bold response sine wave only begins to increase after the PCO2 sine wave starts to increase. The bold response lags behind the PCO2 stimulus. The phase is negative, signifying that the bold response is positive. The gain color scale is based upon whether the phase is positive or negative, and the phase color scales show this aspect too. Here is a phase map from a 44-year-old Moya Moya patient. The histogram shows the distribution of positive and negative phases. Note the color scaling. For a positive response, phase is negative, and the smaller the phase difference, the faster the response. For a negative response, phase is positive, and the fastest response is when the phase is 180 degrees or pi radians. Just in case you are curious about the transfer function analysis calculation, here are some of the details, and you can pause to view these, but I will skip to the results. As these maps from two patients with similar pathology show, transfer function analysis gain corresponds to the ramp CVR, and transfer function analysis phase corresponds to tau. TFA coherence is a measure of how consistent the relationship between the carbon dioxide stimulus and the bold response is over the time of the sequence. So TFA is an alternative analysis that can be used by itself or as a double check of the linear regression CVR measurements. These CVR and TFA maps provide a basic assessment of cerebrovascular health in terms of the ability of the vasculature to increase cerebral blood flow when challenged by an increase in carbon dioxide partial pressure. The conclusion to be drawn from them is that a decreased CVR implies a loss of cerebrovascular vasodilatory reserve. For most patients, these assessments are sufficient for clinical decision-making, and this talk can end here. However, recall the illustrations of the vascular responses to stenosis. It was the vasodilatory reserve that was the important characteristic which determined whether a region could cope with any increase in demand for blood flow or not. To follow that thought, the next section presents a more detailed analysis of the cerebral blood flow responses to a carbon dioxide challenge that can provide an estimate of vasodilatory reserve. While the healthy bold response to a global carbon dioxide stimulus in the top graph is sigmoidal, the blue line, another voxel's response in the lower graph, shown in purple, is not necessarily so. Consequently, a sigmoid does not fit all bold responses to carbon dioxide. A straight line fit, such as the black line in the lower graph, is not necessarily a good description. A better estimate is to separate the response into hypocapnic and hypercapnic ranges, the purple lines, based upon the resting partial pressure of carbon dioxide, shown as the red vertical line. And this approach provides a classification into 4 types based upon the hypocapnic and hypercapnic regression slopes. The 4 types can be used to produce a type map, as this picture demonstrates. Type A regions have a healthy vasodilatory response that is usually sigmoidal. Type C regions show areas where actual steal occurs throughout the range of CO2. Type B regions show a response in the hypocapnic range which fails in the hypercapnic range and steal occurs. It is as if the vasodilatory response has been shifted left to a lower range of PCO2. Type D regions fail to respond in the hypocapnic range and steal occurs, but then have a vasodilatory response in the hypercapnic range. It is as if the vasodilatory response has been shifted right to a higher range of CO2. Note the dotted black lines for the B and D types. They demonstrate the erroneous CVRs that can result by fitting a single regression line to these complex responses, and how the PCO2 range chosen could markedly affect the results. The observed types of bold signal responses to a global carbon dioxide stimulus are the result of changes in perfusion pressure caused by network competition interactions and changes in vessel diameter and therefore vessel flow resistance, and these cannot be separated. Changes in vessel diameter are constrained by the anatomical limits of vasoconstriction and vasodilation. Consequently, the change of resistance with carbon dioxide is sigmoidal. The assumptions for modeling this physiology in terms of competition between regional resistances are as follows. We take the mean arterial pressure as equal to 100 and the venous pressure as 0. Voxel flow is scaled from the bold responses as bold percent of the mean plus 100, and consequently the model units of pressure, flow, and resistance do not actually correspond with real measurements in the brain. The final assumption is that a 30% pressure drop occurs in the major supply arteries from the mean arterial pressure at 100 to P branch, the perfusion pressure. And with the assumption of a mean resting flow of 100 yields an arterial resistance of 0.15 in the major supply arteries. Here is the model of the competition between resistances. The idea is to use a reference resistance, Ref, in one branch of the model to compete with an examined voxel's bold response to a ramp of carbon dioxide, Fvox. This competition simulates the network interactions that change the perfusion pressure, P branch. The model variables that are known quantities are shown in red, and using these values, the resistance model can be viewed as an electrical circuit, and the variables in blue can be calculated. There is a caveat. Note that the actual reference resistance competition for each voxel is different and unknowable. Consequently, the model only calculates the resistance variation with carbon dioxide in the examined voxel that would produce the observed bold response if it was in competition with the reference, not the actual resistance variation with carbon dioxide. In other words, if the voxel blood flow did result from competing with the reference resistance, the model calculates the resistance variation with CO2. By making the reference resistance response to carbon dioxide the same for all observed voxel bold signal responses to CO2, the resistance sigmoid pattern of variation with carbon dioxide can be compared between voxels because it is entirely determined by the measured bold response to carbon dioxide. The model simply converts the bold signal variation in response to the global CO2 stimulus into a more physiologically meaningful sigmoidal resistance form, where the parameters of the resistance sigmoid provide a description of the voxel bold response to a global carbon dioxide stimulus. The resistance sigmoid is still a result of both vasodilation with CO2 and vascular network pressure changes because it is derived from the bold signal response. The resistance sigmoid parameters shown here are as follows. Start is a relative measure of the vasoconstrictive flow limit in terms of resistance. Amplitude, a measure of the maximum extent of vasodilation and constriction flow limits in terms of resistance. Midpoint is a measure of the displacement of the response with respect to CO2, explaining the B and D types of response, as we shall see. Range is a measure of the carbon dioxide range over which the resistance response is linear. Sensitivity at the resting carbon dioxide partial pressure is an alternate measure of the resting state CBR, only in terms of resistance. And reserve at the resting partial pressure of carbon dioxide is a measure of the vasodilatory reserve in terms of resistance. Here is the model solution for a type A voxel bold response to carbon dioxide. The left graph plots flow against partial pressure of carbon dioxide, and the right graph plots resistance against partial pressure of carbon dioxide. The example voxel examined is in purple and the reference is in black. And the solution proceeds by the numbers shown. First, the left graph shows the flow or bold measured data points in purple. The right graph shows the reference resistance line in black. The model solves for each flow or bold data point in the left graph to get the voxel resistance value points on the right graph. These calculated resistance points are fitted with a sigmoid, the purple line in the right graph, for which an R-squared value of the fit is calculated. The model also calculates the reference flow values in the left graph and the sigmoid fit. And it calculates the voxel flow response, which you can see in the left graph is sigmoidal and fits the data points well. And here is the model solution for type B voxel bold response to carbon dioxide. The left graph plots flow against partial pressure of CO2, and the right graph plots resistance against the partial pressure of CO2. And as previously, the example voxel is in purple and the reference is in black. The model takes the voxel measured points in the left graph, converts them to resistance with a sigmoidal fit in the right graph. The voxel resistance sigmoid is then used to calculate the voxel flow response line in the left graph, and as can be seen, the fit is excellent despite the non-sigmoidal response. Note that the midpoint of the fitted voxel resistance sigmoid is lower than the reference midpoint. At the voxel midpoint partial pressure of CO2, resistance sensitivity, or slope, is at a maximum, and so marks the partial pressure of CO2 where regulation is most effective. Has the resting tone adapted to a lower resting partial pressure of CO2 in this region caused by a chronic hyperperfusion? Here is the model solution for a type C voxel bold response to CO2, as shown in the two graphs as before. The model takes the voxel measured points in the left graph and converts them to resistance with a sigmoidal fit in the right graph, and the voxel resistance sigmoid is used to calculate the voxel flow response line in the left graph. And as can be seen, the fit is excellent. Finally, here is the model solution for a type D voxel bold response to CO2. And the graphs are as shown before. Again, the model takes the voxel measured points in the left graph and converts them to resistance with a sigmoidal fit in the right graph. And that voxel resistance sigmoid is used to calculate the voxel flow response line in the left graph. And as can be seen, the fit is excellent despite the non-sigmoidal response. Note that the midpoint of the fitted voxel resistance sigmoid is higher than the reference midpoint. At the voxel midpoint, as we said before, the resistance sensitivity or slope is at a maximum. It marks the partial pressure of CO2 where regulation is most effective. Has the resting tone adapted to a higher resting partial pressure of CO2 in this region caused by a chronic hypoperfusion? The resistance model approach accounts for the four types of bold responses to a ramp of carbon dioxide challenge, resulting from redistribution of flow between vascular beds, and it calculates the pattern of the intrinsic sigmoidal flow resistance changes. It therefore provides an estimate of a physiological parameter, resistance, relative to a universal ideal reference standard, and thus leads to physiological explanations for the observed bold response patterns that we have just reviewed. Given that the shifts in amplitudes and midpoints of the resistance sigmoids occur under pathological conditions, it is likely that maps of these characteristics are of clinical relevance. And here are example maps for a patient, a 56-year-old male with left-sided weakness and right middle cerebral artery occlusion. The maps of the various sigmoid characteristics that describe the resistance change in response to carbon dioxide are as follows. Resistance amplitude, a measure of the full extent of vasodilation and vasoconstriction. Resistance midpoint, the partial pressure of carbon dioxide at which sensitivity is a maximum. Resistance range, the partial pressure range over which the resistance variation can be regarded as linear. Resting sensitivity is the slope of the sigmoid at the resting partial pressure of CO2. It's a measurement analogous to the CVR at a resting partial pressure of CO2. And resting reserve, the vasodilatory reserve available at the resting partial pressure of CO2. This is a direct estimation of vascular health in terms of vasodilatory reserve. And finally, quality maps, the R-squared values of the sigmoidal bits to the calculated voxel resistance data points. The vasoactive stimulus carbon dioxide and the MRI measurement of BOLD for blood flow changes, combined in a simple brain stress test, produce anatomical maps of cerebrovascular reactivity, as well as more complex analytics such as vascular resistance. To be of clinical use, the stress test must have both a stimulus and MRI measurement that is repeatable and precise, so that comparisons between and within individuals may be made, the ZMaps. We have used such a stress test for more than 20 years, and the experience gained is documented in the papers referenced throughout this talk. I therefore suggest that the assessments of cerebrovascular health described here are of clinical utility.